Welcome to Curious Brain. Have you ever wondered what governs the function of a human cell? Let's explore. Chromosomes. The word chromosome is derived from the Latin word chroma and soma. Chroma means color and soma means body. Together, it is called as colored body. What is the definition of a chromosome? A chromosome is a double-stranded, coiled structure which is wound around certain proteins called the histone proteins, which contains the DNA and which in turn contains the genes which controls and regulates the function within the cell. What is the major function of a chromosome? The chromosome carries the genetic material which governs the entire functioning of that particular cell. What are the various types of chromosomes? Prokaryotic chromosome and eukaryotic chromosome. What is a prokaryotic chromosome? What is a eukaryotic chromosome? And what is the difference between the prokaryotic chromosome and the eukaryotic chromosome? A prokaryotic chromosome is single-stranded, whereas a eukaryotic chromosome is double-stranded. A prokaryotic chromosome is found in primitive cells like the cells of bacteria and virus, whereas the eukaryotic chromosome, which is double-stranded and is stabilized by certain proteins, are seen in certain multicellular organisms which has complicated cellular structure and function. How many pairs of chromosomes are there in the human cell? It consists of 23 pairs of chromosomes, out of which 22 pairs are autosomes or autosomal chromosomes, which governs the major function of all the cells in the body, and one pair of sex chromosome. If it is XX, then it is a female, and if it is an XY, then it's a male. Composition. What is the composition of a chromosome? It has DNA, RNA, and proteins. DNA forms the major chunk of the chromosome, whereas RNA helps in protein synthesis and key regulatory functions within the nuclei. The structure of the chromosome is stabilized by histone proteins. Structure of a chromosome. What is the structure of a human chromosome? It is double-stranded, thread-like, and it has two chromatids. What is a centromere? A centromere is the primary constricted area of a chromosome where the mitotic spindles attaches during the time of cell division. What is a telomere? A telomere is the terminal part of a chromosome. As the cell continues to divide, the length of the telomere shortens and has a direct correlation with aging. In addition to the primary constrictions, there are also certain narrowing area in the chromosome called as secondary constrictions. Types of chromatids: sister chromatid and non-sister chromatid. In case of a sister chromatid, the two strands are identical, whereas in case of a non-sister chromatid, the two strands of the chromatid are non-identical. Euchromatin. What is a euchromatin? Any particular segment of a chromosome which is transcriptionally active is called as euchromatin. What does the word transcription mean? Transcription means the formation of mRNA or messenger RNA from DNA. Since euchromatin is loosely bound to the histone proteins, which are the regulatory proteins, it can be transcriptionally active. Transcriptionally active euchromatin helps in the synthesis of proteins. The staining pattern of a chromosome can either be light or dark. Since it is loosely bound, the staining pattern of euchromatin is light. What is the central dogma of the cell? The formation of mRNA from DNA, which in turn helps in the formation of proteins, forms the central dogma of the cell. DNA to mRNA to proteins. Euchromatin plays a vital role in the central dogma of the cell. Heterochromatin. What is a heterochromatin? Any particular segment of a chromosome that is transcriptionally inactive is called as a heterochromatin. What is transcription? Again, transcription is the process by which DNA synthesizes mRNA or messenger RNA. It is tightly bound to the regulatory proteins that are present in the chromosome. What is the regulatory protein that is present in the chromosome? Histones. Since heterochromatin is tightly bound to the histone protein or tightly wound to the histone proteins, it cannot take part in mRNA creation or in protein synthesis. The staining pattern observed in heterochromatin is dark. Autosomal chromosomes. What are autosomal chromosomes? The human cell contains 23 pairs of chromosomes, out of which 22 pairs belongs to the autosomal chromosomes while one pair belongs to the sex chromosome. Any chromosome that is not directly involved with either reproduction or sex determination comes under the category of autosomal chromosomes. Loci. What is a loci? A loci is that particular segment of a DNA where genes are located. In other words, it is the coding segment of a DNA. Genes that are present in autosomal chromosomes are called as autosomal genes. Autosomal genes can either be dominant or recessive. What is an autosomal dominant gene? If the presence of one gene is sufficient for protein synthesis, it is called as an autosomal dominant gene, whereas if two genes are required for the protein synthesis function, then that particular gene is called as an autosomal recessive gene. Sex chromosomes. What are sex chromosomes? The chromosomes that play a direct role in sex determination and reproductive functions comes under the category of sex chromosomes. In humans, chromosome X and chromosome Y come under the category of sex chromosomes. The male karyotype contains the chromosome X and the chromosome Y, whereas the female karyotype contains two copies of the chromosome X. In human beings, there is one pair of sex chromosome while there are 22 pairs of autosomal chromosomes. Together, there are 23 pairs of chromosomes in a human cell. Certain diseases are X-linked. Males are particularly more susceptible to X-linked chromosomal diseases because males have only one copy of chromosomes whereas females have two copies of chromosomes, making the prevalence of X-linked diseases less in females. Chromosomes can be divided into four types based upon the location or position of the centromere in that particular chromosome. Metacentric. What is a metacentric chromosome? If the centromere of that particular chromosome is found at the exact center of the chromosome, then that particular chromosome is called as a metacentric chromosome. Submetacentric chromosome. If the centromere of that particular chromosome is found just above or just below the center of the chromosome, then that particular chromosome is called as submetacentric chromosome. If the centromere of that particular chromosome is found at the end of a chromosome, then that chromosome is called as a telomeric chromosome. That is, it is found terminally at the end of a chromosome. What is an acrocentric chromosome? If the centromere is found near to the end of a chromosome, but not at the end, not at the exact end, but near to the end, then that particular chromosome is called as an acrocentric chromosome. Functions of chromosomes. The chromosomes carry genetic material of a particular cell. The genetic material contains genes, and these genes code for mRNA, which in turn synthesizes proteins. Proteins can either be structural proteins or it can be functional proteins. What is the role of a structural protein? A structural protein gives support through cytoskeleton, whereas a functional protein regulates all the various metabolic activities of a cell. Examples include enzymes. Chromosomes are responsible for the hereditary transfer of genetic material from parent to offspring. What does the word hereditary means? It means the transfer of genetic material from parents to offsprings. The genetic information present in the chromosome is responsible for the maintenance of cellular functions. What is the function of a centromere? During cell division, the mitotic spindles attaches to a centromere during the metaphase stage of mitosis and thereby help in the cell cycle. Chromosomes houses genes which in turn protect it from external damage. Karyotyping. What is karyotyping? The study of the number and appearance of all the chromosomes of a particular eukaryotic cell in a species is called as karyotyping. The human karyotype consists of 23 pairs of chromosomes, out of which 22 are autosomal chromosomes and one pair is the sex chromosome. How is karyotyping done? The cell is arrested at the metaphase stage of mitosis through the substance colchicine, and then it is observed under the microscope. What are the uses of karyotyping? It can be used to detect gross genetic abnormalities like monosomies or trisomies, which is nothing but increase or decrease in the number of chromosomes. It can also be used to study the entire genetic composition or the genome of a particular species. An important application of karyotyping is its detection of chromosomal abnormalities. The most common chromosomal abnormality is Down syndrome. What is Down syndrome? Here there are 3 copies of the chromosome 21. Normally there are only 2 copies of the chromosome 21. If there are 3 copies of the chromosome 21, that is called as trisomy 21, which is otherwise called as Down syndrome. Down syndrome is usually associated with increased maternal age. That is, the greater the age of the mother at the time of conception, greater is the risk of that particular mother's baby developing Down syndrome. Some traits of a Down syndrome baby include flat face with Mongolian features and low set ears. Intellectual disability is usually permanent, and developmental delays are also observed in that particular baby. There is no specific treatment as of now, and genetic counseling before time of conception is usually preferred in order to prevent conception with Down syndrome. We have come to the end of the segment. I hope you like this video, and if you like it, please hit the thumbs up button and consider subscribing to stay tuned to A Curious Brain to get informed about various interesting scientific content. Thank you.